Okay, this is the cell cycle, which is also called mitosis, which happens to be somatic cell division, body cell division. So mitosis just deals with the cells of the body, not the reproductive cells. We'll talk about that later. Now, binary fission. Binary fission is a simple type of cell division in prokaryotes. So this is how bacteria divide. It's by binary fission. And you can see it's pretty fast. Some can do it every 20 minutes. So bacteria can divide very rapidly. They carry out all their cellular metabolic processes, get large enough, they can divide, and some can do it every 20 minutes. That's a tremendous feat. Not all of them do it every 20 minutes, but this just lets you know that bacteria can reproduce quite rapidly, and when you have a bacterial infection, you want to be sure you get on top of that quickly with taking extra antibiotics. Here is just a little diagram showing binary fission. And one thing about prokaryotes, if you remember, they have one chromosome and it's circular. And that chromosome you see is attached to the plasma membrane on the inside. As it is replicated, it's copied, and then the cell elongates, which picture 2 doesn't really show elongation too well. But as it elongates, the 2 strands are pulled apart. The 2 circles are pulled apart. And a cell plate starts to form in the middle until it separates the 2 into identical clones of each other. So when binary fission occurs in bacteria, they produce clones of themselves. And it's an exponential thing. They double every so many minutes or hours. The numbers of bacteria will double, and those are all clones. They're identical. Now we have a term, a genome, an individual or group's genetic makeup. The Human Genome Project took place, I don't know, 10, 15 years ago, and they figured out the genetic makeup of humans. Now they don't know what all the genes are, but they know what the code is. And so now they have all this human genome, and now they got to figure out what all these genes do, how they affect the cellular activity. They're involved in different things, the heart, the stomach, development, aging, whatever. That's what scientists are doing now, is figuring out what each of the genes function happens to be. And every so often you'll hear about this on the news, that they found the gene for a particular trait or problem or function. Well, that's good, but there are a lot of genes to figure out. So every one they figure out, though, is more knowledge, and it helps us to possibly solve problems in the future. Now we have the entire genome of E. coli. That's a bacterium, E. coli bacteria. The fruit fly, those are the Drosophila melanogaster. Those are the ones you bring in with your bananas and fruits from the grocery store. And C. elegans is a type of a roundworm. It's a real small little roundworm. But we know the entire genome, and we know exactly what all the genes do in their genomes. Now that's pretty good, because over the years, things have happened in science, and one thing that's happened is called recombinant DNA, where DNA can be recombined with different, let's say, species or different organisms. And that's what you call GMO, genetically modified organisms, is due to recombinant DNA. Well, sometimes that's not good. It could be used for bad purposes, but what we've done with a couple of things has been pretty beneficial to man. In the past, diabetics used to have to get insulin from pig pancreases, because pigs have a lot of genes that are similar to humans, and the insulin was very similar, and it worked in humans. And so they would get pig pancreases, grind them up, distill out the insulin, but to give for their insulin injections. But they also would bring over some proteins that were pig proteins that would be mixed in with the injection. And so people were making up antibodies against those foreign proteins and starting to have reactions to the insulin injections. Well, due to recombinant DNA, scientists found a way to clip out the human insulin gene and insert it into these E. coli bacteria. And so now we have a special subspecies, I guess, or genetically modified species of E. coli bacteria that will make human insulin. And so they put these bacteria into a big vat, warm it, has nutrients in there, paddles, it's spinning around, churn it around, warm it up for a given amount of time. They turn it off, kill the bacteria, filter them out, and they can get the insulin out of the mixture very easily. And it's human insulin that we use for injections now, but it's made by E. coli bacteria. So knowing what genes do, the more we know, the more we can solve problems or come up with beneficial combinations, for crops being more resistant, more productive, this type of stuff. So definition, division of the nucleus. That's what mitosis means, is division of the nucleus. But we use it to represent the concept of somatic cell division, body cell division. So mitosis is the division of the nucleus to produce two genetically identical daughter cells. So mitosis is a cloning of the parent cell. And here I have a parent cell here that has two chromosomes, a red and a black. Well, so do the daughter cells. They have one red, one black each. So they have the same genetic composition of the original parent. Now, the second part of this mitosis is cytokinesis. This is division of the cytoplasm between the two new daughter cells. And so the daughter cells have the exact same amount of DNA as the original parent, but roughly 1/2 the amount of cytoplasm. So they're roughly 1/2 the size of the original parent, but they have the exact same size nucleus as the original parent did. Some forms of DNA. In a cell that is not dividing, the DNA is in long, thin strands called chromatin. See, it says unwound DNA. It says DNA, RNA, proteins. They're long, thin threads of DNA found in non-dividing cells. If a cell is not dividing, the DNA is in the form of chromatin, long, thin threads of DNA. And that allows all the genes to be accessible for whatever the cell needs. Whatever it needs, the gene is accessible, and it can acquire the information off of the DNA to make whatever polypeptide the cell needs for survival, growth, and repair. Now, chromosomes are another form of DNA, tightly coiled DNA. So when a cell is going to divide, the chromatin is coiled up into short visible units called chromosomes. So it says tightly coiled DNA, the genetic material of the cell, visible only during mitosis. You're only going to see chromosomes when the cell is dividing. That's the only time you will see chromosomes, is when the cell is dividing. Otherwise, the DNA is unwound into long, thin threads of DNA called chromatin. Here's another definition, chromatids. Now, in mitosis, cell division, the chromosome replicates, it copies itself, and is now composed of two identical strands of DNA called sister chromatids. Now, they're called sister chromatids because they are together. So you see my chromosome's kind of rough here. It replicates, it copies itself, and now there are two sisters. The blue arrows are pointing to the two sisters. And they're held together in a region called the centromere. So since they're held together in this centromere region, we're going to see that there's something inside of there. There's a protein that holds those two together, and it has a specific name. Since it's still one structure, it's called one chromosome. It's like if you get your highlighter, the highlighter has a cap and the main portion. If you have the cap on there, that is one piece, and if you toss it around the room, it's one piece. But if you take it apart, take the cap off, then it is two pieces. The same thing right here. As long as these two sister chromatids are held together in this centromere by a structure we're going to see in a minute called the kinetochore, holds them together, it's still one structure like your marker. And until those two sisters are separated, they are one chromosome. When they're separated, each one then will be a chromosome, but not until they're separated. And that's called a dyad. Two sisters is called a dyad. Di is two. Now here on the far left, you see a chromosome, and you show it's replicated, that little image there. And you come right over to the middle, you see an electron micrograph. That's going to be a scanning electron micrograph because you see nice 3D surface detail here. And you see it is held together, just like the drawing on the right. And the region in the middle, the region that they're held together in, the region or area is called the centromere. But the structure in that centromere that holds them together is a protein called a kinetochore. So those two little white dots, those are the proteins, the kinetochore holding those two sisters together in the centromere, that region. Otherwise, you have your two sister chromatids here, and that's one chromosome. Kinetochore definition. It's a structure, it's a protein, which holds the two sister chromatids together. It says DNA and proteins. The DNA is in the chromatid, and the proteins is kind of intermeshed there with the DNA. So here are the two sisters again. Here's the centromere. A region or an area is the centromere. That's not a structure, it's a region. The kinetochore is the protein that's holding those two sisters together. It's a physical structure that's inside of this region called the centromere. So that's holding those two sisters together, and until that kinetochore is split, those two sisters are held together as one chromosome. Now there are some microtubules. This kind of jumps a little bit ahead, and we'll see on another picture how these microtubules are positioned, but this kind of gets ahead just for a moment. Function in structure and movement in cells. Well, you knew that. You knew that microtubules were involved in cell shape and movement inside of the cell. The microtubules which attach to the kinetochores of the two sister chromatids are called Kinetochore Microtubules because they attach to the kinetochores. And you're going to see in a minute that they're coming from poles, opposite sides of the cell. And I've got my sister chromatids labeled here, and I have the microtubules that are attaching to the kinetochore, and they're called the Kinetochore Microtubules. They move the chromosomes to the equator of the cell, and they will eventually pull the sister chromatids apart. Okay? The microtubules they're called spindle fibers. They're part of the spindle fibers, the spindle apparatus that we'll see on the next slide. So, in mitosis, we're going to see some events are going to occur, and we will see that this little cage is set up. Centrioles are going to move to opposite poles, like the Earth has a north and a south pole, and as they do that, the microtubules are laid down between one set of centrioles and the other. These microtubules are called, well, they're part of the spindle apparatus, okay, and at the end here, you can't see it too well here, but you can at the bottom of this picture, and you can over here, where the microtubules anchor the apparatus, to anchor the cage, is called an aster. They kind of fan out like a little starburst and anchor this spindle apparatus, this cage of microtubules. Some microtubules attach to the kinetochores, and when they attach to the kinetochores, they're called Kinetochore Microtubules. Ones that do not, they go from one pole to the other pole, and they're called non-kinetochore microtubules. Now they have overlapping on this one for some reason, non-kinetochore microtubules. So non-kinetochore go from one pole to the next pole. Kinetochore microtubules go from one pole to one side of the chromosome and attach to the kinetochore on this side, and from the other pole and attach to the other side. And you can see how these have been lined up on this picture on the upper right. Lined up along the equator, you can see the cage, the spindle apparatus, you can see the asters, and these are lined up. The bottom right picture, which we have another one on the next page, but this is a really big blow up. Here's a kinetochore microtubule on this side of the kinetochore and one on the other side of the kinetochore. So the dark mass in the middle, that's the kinetochore holding those two sisters together. This is blown up quite a bit larger. This is the kinetochore microtubule from, like say, the North Pole, and here's one from the South Pole, and they're attached to the kinetochore. And you can see some of the arms of the sister hanging off to the side, the two sisters. So kinetochore microtubules attached to the kinetochores of the sister chromatids. Non-kinetochore microtubules do not attach to kinetochores. That's why they're called non-kinetochore. They extend from one pole to the other, the poles being the opposite sides where the asters are and the centrioles are. Now here's what they do. They cause the cell to elongate to help separate the two sisters. Now, one thing about the non-kinetochore microtubules, at a certain time in mitosis, they will elongate to make the cell kind of become a little bit more oval. Well, the kinetochore microtubules do not elongate. And so when the spindle apparatus elongates, it's going to put stress on those kinetochore microtubules, which are going to put stress on the kinetochore and will break it in half, pull it, split it in two. And so it'll separate the two sisters at that point. That's how they get separated. Here's just the picture again. You can see the centrioles at the top and the bottom. So those are the poles. You got a good picture of the aster. You have these starburst out at the different poles to anchor the spindle apparatus, which is all the microtubules going between the poles. You see I have circled overlapping non-kinetochore microtubules. I just put, it's just for the non-kinetochore microtubules. I don't know why they put them overlapping like that, but they go from one pole to the other. And then on the bottom right, you see kinetochore microtubules go from one pole to one side. That's that kinetochore on the bottom, that part of it, from the north pole to the other part of the kinetochore on top. So you can see that the kinetochore has two parts, and it can be split. But it only can be split if they're being pulled on both sides, which will happen at one phase of mitosis. Some more new terms. Homologous. Homo means same. So these are the same or like. It means that they contain the same genes for the same traits. Now you get chromosomes from mom and chromosomes from dad. They're in the eggs and the sperm. Mom has 23 chromosomes and they're numbered 1 through 23, and dad has 23 chromosomes and they are also numbered 1 through 23. Number 1 in mom and number 1 in dad have the same traits for the same types of characteristics. So those are homologs. Number 7 and number 7 for mom and dad, they both have the same genes for the same characteristics. But number 1 and number 7, they do not have the same genes for the same traits or characteristics. And so those are non-homologous chromosomes. So they are numbered for a reason. They have different lengths and different banding patterns, so they can be paired up pretty easily on a picture. But homologous means they contain the same genes for the same traits. So number 1 pairs with number 1, number 2 with number 2, number 3 with number 3, and so on. So it says they contain the same genes for the same characteristics, although the genes may not be for the same expression of that characteristic. And some examples are eye color. There are different eye colors. So there's a gene for eye color, and this, not unless this PowerPoint is going to work or not, but A was for eye color, and you can see that mom has a large gene, a large A for her eye color, and dad has a small a for his contribution. And so the offspring will have a particular eye color. It could be mom's or dad's or a blend. Skin tone, you have light colors and you have darker colors. Now, eye color and skin tone, those have more than one of these little alleles, which are these coding units here called the big B and the little b. So mom had the big B, dad had the little b, and we'll see what those stand for later. Finger length, short fingers, long fingers, middle. Tongue, that could be a D, they could both be tongue rollers. So all their kids are going to be tongue rollers. Or one could be a tongue roller with let's say, a big E and a non-tongue roller with a little e. Well, that's what my family is. I'm a tongue roller, my wife is a non-tongue roller, but all my kids can roll their tongues, okay, because I gave them the tongue rolling characteristic. But they all have one dominant and one recessive gene for that. We'll get into those later. So homologous chromosomes contain the same genes for the same characteristics. Homo means same. Just like homosexual means same. 2 males or 2 females. A heterosexual means a male and a female. So you're probably more familiar with those terms, but homo means same, hetero means different. Now somatic cells, the word soma means body. And so somatic cells are body cells. Like bone, like I have right here, skin, muscle, nerve, bone, blood, those are body cells. Have nothing to do with sexual reproduction. Nothing to do, because those are going to be gametes. Gametes are sex cells, and sex cells are your ova, the eggs, and the sperm, eggs from the female, sperm from the male, involved in sexual reproduction. Gametes only have, each one of them has one set of chromosomes. The ova have a set from 1 to 23, and the sperm have a set from 1 to 23. So they're what we call haploid cells. They only have one chromosome, which let's look on the next slide or two and we'll still see that. The diploid number. Di means two, ploid is sets, two sets. You got one from mom, one from dad. So it's a full set of chromosomes. Di means two, ploid means sets, there you go. The human diploid number is 46 chromosomes. In humans, it's called the 2N number, that's the diploid number, is 46 chromosomes or 23 pair. Because just like you have a left and a right shoe, that's a pair of shoes. You got a chromosome from mom and a chromosome from dad, that's one pair. So number one, it's a pair. Number two, it's a pair all the way to 23. So 46 chromosomes, 23 from mom, 23 from dad, so you have 23 pair. My example here doesn't have 46 chromosomes in it because I'm not going to draw 46 different shapes on here. So I made up my play cell, and my play cell, the diploid number, has 6 chromosomes. I have 2 greens, I got one from mom and one from dad. 2 reds, got one from mom, one from dad. And 2 blues, got one from mom and one from dad. That's the diploid number for this cell that I made up. It only has 6 chromosomes. The 2N number is 6. That's the diploid number. So this is what all the cells of that organism's body would have, would be 6 chromosomes. Now the haploid number, 1N, is haploid. Remember 2N was diploid, 1N is haploid, it means one set. This is how the haploid number is written, 1N or just N. The haploid number is 23 chromosomes. This is 1/2 the full diploid chromosome number. So in this example, the diploid number was 6 on the previous cell, right? Well, the haploid number is 3. So N is 3. This is what an egg or a sperm would have. One green chromosome, 1 red, and 1 blue. That's the haploid number. That's what your eggs and sperm would be like. They have 23 chromosomes each. And when fertilization occurs, when the sperm combines this nucleus with the nucleus in the egg, that's 23 plus 23, that's 46. So the diploid number is reestablished at fertilization. Now polyploidy, poly means many and ploid means sets. This is many sets. More than a full set of chromosomes. That's true. An example would be triploid, quadruploid. Triploid has 3 sets, quadruploid has 4 sets. Animals cannot deal with multiple sets. Diploid is what animal cells can handle. Triploid, no, they die. Quadruploid, they would die. Some plant cells can live with multiple sets. Some plant cells, or plant organisms, can live with polyploidy. Strawberries are octoploid. They have 8 sets of chromosomes, and they are fine. I think lettuce is another one that's polyploid. So it happens in plants and they can live. Animal cells do not tolerate multiple sets. Not multiple sets. Now we have something called the cell cycle. The cell is going to be in one of these parts of the cell cycle at any one point in time. It's never not in one of these parts of the cell cycle. So the cell doesn't rest. Like when I was younger, they told us the interphase, it was a resting state for the cell. It's doing anything but resting in interphase. So we're going to see a thing called the cell cycle, which is a drawing that shows, the events in the life of a cell. And it includes the events of interphase, which we will define G1, S, and G2. Those are parts of interphase. That's when the cell is not dividing. It's called the vegetative state. It's not dividing. And then mitosis, the cell is starting to divide. And so we have some phases there, prophase, metaphase, anaphase, and telophase, and we'll go over those. So here's the cell cycle. Here's G1. Here, see all this is interphase here. This is when the cell is not dividing. And in G1, we'll talk about that. Yes, and G2. Now, when the cell is starting to divide, we know it's going to divide in prophase. Certain events happen, and this is all red. So prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis, division of the cytoplasm, and here's your two new daughter cells. Once they are formed, each one of those enters into G1. After cytokinesis, they both enter into G1, and the whole process continues. So the cell is constantly in one of these states of the cell cycle, one of these parts of the cell cycle. So let's look at what happens to this daughter cell after it's just been formed. It enters into G1, which is the first growth phase. That's what G1, growth 1, first growth phase. The cell is diploid because remember mitosis, all the somatic cells are diploid. They have a full chromosome number. The part of interphase, the new daughter cell enters after it is formed. So that's the first part it enters into. It goes straight into G1. Because the cell is small, and in order to divide, the cell has to be a certain size. If a cell divides and the daughter cells are not large enough to support life, they die. And so the purpose right now, G1, is to start this cell growing to get large enough to increase its biomass big enough to where it can divide and both daughter cells can survive to do it again. So the cell has to grow, and in G1 it's going to be producing proteins, carbohydrates, lipids, and nucleotides. Remember what nucleotides are? They're the building blocks of DNA. So it's not building DNA in here, it's making the building blocks, as well as enzymes, which are part of the protein component there, are being made. So it's growing its biomolecules and growing the building blocks for replicating the DNA. So it's trying to increase itself in size. So nucleotides are synthesized, not DNA. Synthesizes enzymes needed to duplicate or replicate the DNA are also done in here. So carbohydrates to increase in size, proteins to increase in size and function, lipids that it needs, and the building blocks and enzymes to make or replicate the DNA. That's what G1 is all about. The S phase, S stands for synthesis. This is where the DNA is replicated, is copied. All those nucleotides that were made previously in G1 and the enzymes are used to copy the DNA. So the DNA is replicated, and all the chromosomes now consist of two sister chromatids held together by a kinetochore in the region called the centromere. So they've all been replicated, and each chromosome consists of two sister chromatids. G2, the cell is continuing to grow, keeps growing, more biomolecules, carbohydrates, lipids, proteins, but in here it's making enzymes to support mitosis and microtubules to form the spindle apparatus. So it's making tubulin to support the spindle apparatus for mitosis. So it's growing to get large enough so that it can divide and produce two daughter cells right before mitosis starts. So it's got to complete its growth of everything it needs for biomass, as well as everything that's going to be needed for mitosis. Because once the cell enters into mitosis, there are no genes accessible. So all the genes are closed. Remember, the chromatin is going to coil up into visible units called chromosomes when the cell divides. And at that time, the genes are now no longer accessible. So right now in G2, the chromatin is still long, thin strands of DNA called chromatin, and all the genes are accessible for whatever the cell needs to make to grow and to make everything that's needed for mitosis. So here we have the phases of animal cell. I just put them on here. Interphase, you see the DNA over here is all unwound in long, thin strands, and there's your nucleolus where you got some protein activity, RNAs going on in there, making proteins. Here are your centrioles at the top, see as pairs. In prophase, things start happening. The centrioles are going to start moving to opposite poles, laying down their microtubules to form the spindle apparatus. The nuclear envelope is going to start to dissolve. You see little holes in there. Well, it hasn't dissolved yet. It's dissolving over the next one. You see the chromosomes are starting to be visible. So they're starting to coil up. Now the DNA, the genes on the DNA are no longer accessible. And prometaphase, which used to be called late prophase, you can see that the spindle apparatus has been laid down. The nuclear envelope is dissolving, continuing to dissolve. The chromosomes are continuing to condense, become visible, shorten and thicken, and you see that the kinetochore microtubules are attaching to the kinetochores and starting to move them around inside of the cell. Metaphase, you see that the chromosomes are going to be lined up, and there's your kinetochore microtubules from one pole to the other, and there's even your non-kinetochore microtubules there. Anaphase, we're going to see what happens. They're going to the non-kinetochore microtubules elongate. You can see this cell is a little bit more oval here than the previous one, whereas the non-kinetochore microtubules do not elongate. And so as the cell elongates, there's stress put on those kinetochore microtubules, and the kinetochore is split, broken in half, and now the sisters are separated and moved to opposite poles as chromosomes. We see something right here. It's called a cleavage furrow, and that means that cytokinesis has started. And once you see the cleavage furrow and know that cytokinesis has started, the cell is in telophase, and that will result in producing 2 daughter cells when that's complete. This is an animal cell showing the same thing. This is interphase. Well, this is actually prophase here because you can see some granules starting to form here. Yeah, prophase. Nuclear envelope is dissolving. You can see the chromosomes real well here in prometaphase. You can't see the spindle apparatus, but it has a spindle apparatus here that's being laid down. You can see it better down here on metaphase when the chromosomes are lined up in the middle. Now, one thing about plant cells is they do not have centrioles. So the centrioles are not at the poles. There are no centrioles there, but they still form microtubules from one pole to the other, from one side to the other. And metaphase, the chromosomes are lined up in the middle. Anaphase, they've been pulled apart, and the sisters are grouping at opposite poles, going to form a new nucleus. Telophase, you can see what's called a cell plate being formed here, and that indicates cytokinesis has started, and you know the cell is in telophase. And eventually, when that cell plate is completely formed, you'll have 2 daughter cells, which are exactly identical to the original parent. They have the exact amount of DNA, same size nucleus as the original parent, but are roughly 1/2 the size. And you can see here was what the size of the original parent was, and now it's going to be half. So the cytoplasm is divided between the 2 new daughter cells. So we have a plant cell here that's in prophase here. You can see that the chromosomes are starting to condense here. So it says chromosomes appear under the light microscope. That was the first description of this process occurring. And so when you see chromosomes starting to condense, shorten, and thicken, the cell is in prophase. Centrioles are going to start migrating. You can't see them on here, but in this case, the reason you can't see them is because this is a plant cell. Plants do not have centrioles, but they're still going to form the spindle apparatus from one pole to the other. You're still going to see microtubules. So it says centrioles separate in the cytoplasm, forming a spindle apparatus only in animal cells. Plant cells do not have centrioles. There's still going to be a spindle apparatus formed, but no centrioles. So that's prophase. Here's prometaphase. You can see that it's still somewhat of a nuclear appearance here, although the membrane's being dissolved. And here are the chromosomes. You can see them real well. Here's the nucleolus, which is going to dissolve. And so it says nuclear envelope fragments, chromosomes thicken, kinetochore microtubules, which you cannot see on this picture, from each pole of the cell attached to the kinetochores at the centromere of each of the chromosomes. So the kinetochore microtubules are going to attach to the kinetochores of these chromosomes and start moving them around. Chromosomes move about by jerky movements. You've probably seen time-lapse photography, and those are taken every, so many minutes or whatever. And it is a jerky movement, but you can see things are changing. In the nucleolus, this is going to disappear, it'll dissolve out in prometaphase. Now, the mitotic spindle here, this is showing another animal cell here because it has centrioles. Again, you can see the spindle apparatus being set up. There's your aster there, your kinetochore microtubules, your non-kinetochore microtubules, and a real picture over here showing the asters. You can see them real well. You can see the microtubules going from one pole to the other, and you know that some of those microtubules are attached to the kinetochores because they're all lined up here along the equator. Here's that picture we saw a minute ago, kinetochore microtubules attaching to the kinetochore. And here you go. You can see the spindle apparatus here. You can make out the shape of tubules going from one side to the other. But this is in metaphase. It means midphase. The chromosomes are in the middle or the equatorial plate of the cell between the poles. So the centromeres are pulled to the equatorial plane, the metaphase plate in the middle of the cell by the kinetochore microtubules. So they're lined up in the middle. Sister chromatids are attached to the microtubules from opposite sides. So these are still made up, each chromosome is still made up of two sisters. They haven't been separated yet. Now we're in anaphase. Centromeres are, well, it says centromeres of the sisters are split. Yeah, the kinetochores are split. And the sisters are pulled to opposite poles. So it says sister chromatids are now chromosomes, are pulled to opposite poles of the cell by the kinetochore microtubules. So there's your spindle apparatus still set up here, but the kinetochore microtubules are pulling the new chromosomes to opposite poles. And you're going to have a nucleus form around that DNA. And then next phase will be telophase. Here is telophase. It says non-kinetochore microtubules elongate the cell. That's what caused the sisters to be split. Daughter nuclei to begin to form. So you can see the daughter nuclei are forming. Here's what's left of the spindle apparatus going across here. Chromosomes uncoil. The nuclear envelope forms. It's going to form a nuclear envelope around here. The DNA is going to unwind back to the chromatin state. And in telophase, you know you're in telophase because you see cytokinesis occurring here. Usually cytokinesis occurs by the formation of a cell plate in plant cells. And we saw that it was a cleavage furrow in animal cells. So when you see the cell plate in plant cells, you know you're in telophase. When you see a cleavage furrow in animal cells, you know you're in telophase. Here's some animal pictures. This is showing interphase, kind of grainy in here. So you see this all unwound chromatin. There's your centrioles getting ready to move. Here's prometaphase. You see that nucleus. Now you see some structure starting to form in here. It's not grainy anymore. You're seeing some solid structure starting to form. You can kind of make out that the spindle apparatus is starting to be laid down here. So it's showing the centrioles moving into opposite poles, laying down the spindle fibers. Some of those are going to start attaching to the kinetochores. Prometaphase, a little further along. Still on this one you can't see too well. On this cell, I don't know. We'll see it later. But the spindle apparatus has been formed. You can see that the chromosomes are more visible here than they were in the prometaphase and the prophase. Now they're in prometaphase. You can see them better. Metaphase, chromosomes are lined up along the equator. There's the asters at both poles. And you can see the spindle fibers going from one pole to the other, just like this drawing shows. So they're lined up. So the kinetochore microtubules have moved the chromosomes to the middle of the cell between the poles, the equatorial plate, metaphase plate. Anaphase, the sisters are being pulled apart and now are moving to opposite poles as chromosomes. That's a good picture there. This is a good drawing here showing that same event. You know you're in telophase. See that cleavage furrow where it bends in? And you can see it's kind of being pinched in two. Microtubules are going to form a ring and pinch that in two to separate the cytoplasm. And there's the cleavage furrow being pointed out here, and you can see it on the top picture here and here. And that'll eventually separate it into two daughter cells. So each of the daughter cells is going to have a nucleus formed around the DNA for the two new daughter cells, which will be identical to the original parent. Remember, they have the same amount of DNA, half the amount of cytoplasm. Here is another picture showing phases of animal mitosis, just like you saw previously, and it goes into a little explanation for each one, so you can read through that. The pictures here are good. They're very good for showing what you need to see on each of the slides here. So you see in the early prophase, you can see that the chromosomes have started to become condensed or visible. Nuclear envelope is starting to dissolve. Late, you can see that the spindle apparatus has been set up over here, and the kinetochore microtubules are starting to move the chromosomes around. Metaphase, chromosomes lined up at the equatorial plate between the two poles. Kinetochore microtubules have them lined up there. When the non-kinetochore microtubules elongate the cell, see this oval here, it causes the kinetochore microtubules to pull the kinetochores apart. And now the sisters are separated and they migrate to opposite poles as chromosomes. Well, you know that the cell is in telophase when you see the cleavage furrow. Otherwise, it's still in anaphase. When you see the cleavage furrow, you know it's in telophase, and the cytoplasm is going to be divided between the two new daughter cells. You see the nuclear envelopes reforming around the DNA. They're going to unwind back into the chromatin state. Here's a scanning electron micrograph showing this cleavage furrow starting here on an animal cell. So that's a pretty good shot there. Cleavage furrow starts and it pinches the cells into two new daughter cells. This is a cell plate on a scanning electron. This is a transmission. This one doesn't show any depth. It shows a cell plate being formed. Here it is being formed, so you know it's in telophase. Further formation, and then all the way forms two new daughter cells. Same size nucleus, approximately 1/2 the cytoplasm for each cell. This slide's just showing, you don't have to look at these three over here, just look at this upper left one. It's showing the binary fission that happens in bacteria. We're not getting into any of the other abnormal type mitotic processes. Now, cyclical regulation. What regulates that cell cycle? How does the cell know to go from interphase into mitosis? Well, we got to look at some things and then we'll look at a picture. So we have protein kinases. Remember what a kinase is? Enzyme that breaks down ATP to phosphorylate something, to phosphorylate a protein. So promotes the activity of other proteins by promoting the transfer of a phosphate from ATP. Remember, kinases break down ATP to phosphorylate some molecule. That's going to activate it, right? Cyclins. Now, cyclins are a reflection. That word means like of a cycle. So it says cyclins change the activity of the kinases. Concentrations of cyclin fluctuate around the cycle. That means sometimes it's going to be present, sometimes it's not. The cyclins, by being present or not, are going to regulate the activity of a particular kinase, a protein kinase, called cyclin-dependent kinase, CDK. That means that cyclin is dependent upon kinase. Now we have a third thing, maturation promoting factor. Maturation promoting factor. To mature is to complete something, like your life cycle from being conceived to growing to dying. That's your maturation part of your life cycle, your developmental pathway. That's what's happening here. This maturation promoting factor is going to send the cell cycle into mitosis to complete the cell cycle back around to interphase again. Maturation promoting factor complex of CDK and cyclin. So they're going to be put together. Cyclin-dependent kinase and cyclin. Promotes the move from interphase to prophase. So that's what causes the cell's cell cycle to go from interphase, where it's been growing, replicating the DNA, growing and making everything. It sends it into cell division. At the end of mitosis, the maturation promoting factor, that complex that sent the cell cycle into mitosis, switches itself off by activating a protein that destroys cyclin. And now the cell will go back into interphase. The daughter cells will enter into interphase. So here is the cell cycle written a little bit differently. And you can see that cyclin-dependent kinase is always there. But it's a cyclin that's dependent upon, it's a kinase that's dependent upon cyclin. Now, look at what happens. Interphase is going along, cyclin-dependent kinase is there. Oh, CYCLIN turns up. And when CYCLIN binds, it makes the maturation promoting factor and it pushes it out of interphase and into mitosis. Now G1 made everything that was needed. It was just waiting. Now, something I didn't tell you was that at the end of G1, everything is checked. The cell is constantly checking and making sure everything is like it's supposed to be. And so at the end of G1, it's checking. It says, okay, am I the right size? Yes, I got all the proteins and lipids and carbohydrates I need. Yes, made all the microtubules. Yes, made all the enzymes needed for mitosis. Yes. And once all those are off of its checklist, the last order on that checklist is to make CYCLIN. And when it makes CYCLIN, that's what pushes it out of interphase and into mitosis. So the last order is to make CYCLIN. And that's where it's coming in here, binding with the cyclin-dependent kinase, which moves it from interphase into mitosis, and the events of mitosis carry on. At the end of mitosis, there's an enzyme that's produced, which destroys the cyclin, and the daughter cells enter back into G1 of interphase. Cancer facts. Cancer escapes the control of cell division because cancer cells are, somatic cells that have undertaken abnormal cell divisions. That's called cancer. So they escape the control of cell division. They ignore density-dependent inhibition. Density-dependent inhibition is where if a cell is next to another cell, it will not divide. That's why you have skin, and this skin is right there. But if it gets cut, well, there's a vacancy there. Now the cells are not next to each other, and cells start to divide to repair that wound in between. And when they touch, they stop dividing, and the repair is complete. So cancer cells don't necessarily obey the density-dependent inhibition, and they can form masses of tissue we call tumors. Some of those tumors are just masses of tissue that kind of crowd out the healthy tissue, and other is masses of tissue that replace the healthy tissue. When they replace the healthy tissue, then the healthy tissue is no longer there to function like it needs to, and that's one of the things that causes death. Cancer cells may stop the cell cycle at any point. So any part of the cell cycle could just freeze for an unlimited amount of time or just permanently. But the cells can divide indefinitely. That means that they're immortal, caused by changes in genes that control cell division. What happens in cancer cells is that a gene called telomerase, the telomerase gene, is turned back on. Here's what that does. When your cells divide, there is an enzyme called DNA polymerase. It makes a polymer of DNA. Well, it locks on to at the end of the chromosome that has what's called a telomere. And when it locks on, it copies from that point down. And so where it locked on, that part's not copied, but from that point down is. And so every time the cell divides, the telomere gets shorter and shorter and shorter, and that's considered to be part of our aging process. Well, cancer cells turn that gene back on. And so every time the DNA is copied and the next strand is copied and is shorter, telomerase adds the telomere back on. And so every time the gene is copied, the DNA gets shorter, telomerase adds the telomere back on. And so the gene never gets shorter. The telomere is always repaired. And so these cancer cells can live indefinitely. There are some cells called HeLa, H-E-L-A, stands for Henrietta Lack. She was a lady back in, I think, 1951, that had, I think it was ovarian cancer. And they actually collected some of the cells, and that was the first cancer cell culture that grew. And it is still alive today, and they still do research with those cells, and they're unchanged from when they were collected back in around 1951. So they are pretty much immortal. Now tumors, they're masses of cancer cells. And there are several types. A benign tumor, benign, means good or well. These are non-life-threatening. Malignant, mal, means bad or fight. These are the bad ones. These are the ones that can kill you. Now, that doesn't mean that benigns can't become malignants, because they can. So if you go to the dermatologist and they says, oh, that's just a benign tumor, it's okay. You want to have them take it off anyway. I mean, you're there, you're paying them to do something while you're there, have them take it off. Benigns can become malignant. Not always, but they can, and sometimes those will end up killing you. Malignant cells have an abnormal number of chromosomes, abnormal metabolism, and do not function normally. Okay, so they're not going, when they replace tissue, they're not replacing that tissue with healthy functional tissue. That tissue no longer functions like it's supposed to. There's a type of cancer called metastasizing cancer. A metastasizing cancer is one that spreads. This is, if you know someone that has a metastasizing cancer, they've got like a 5% chance of living. So there is still a chance of living, but they got a 95% chance of dying. What metastasizing cancers do is they spread. These are the ones that you hear about. Someone has kidney cancer and they go in for chemotherapy and they get it taken care of, and now they got liver cancer. And while they're doing liver cancer treatments, they get stomach cancer, and then it goes to brain cancer. You know, it just travels around the body and they eventually die. I lost my Uncle Bob, by metastasizing cancer. It went from his lungs to his stomach, to his pancreas, to his intestines, to his brain. He tried to fight it as long as he could, but it eventually killed him. But there is a 5% chance of surviving, a 95% chance of not surviving. Here's a scanning electron micrograph. Again, it's a blow up again of a plant cell. And there's a cell plate in the middle. It's going to form, it'll eventually become cellulose to make a new cell wall between the two daughter cells. If you were in lab, you would have seen these pictures here. This would have been the animal cell slides you would have seen. They're not quite as pretty as the onion root tip here, where you can see some nice chromosomes. They use different, there's metaphase right there. Probably prophase, it's going to be interphase. Those are daughter cells, new daughter cells, interphase. That's definitely probably a metaphase there. So here's either an anaphase or a telophase right there at the top. So you know, you could have seen some of those, and I'm sure in your lab there's some pictures. Okay, so this is the end of your for cell cycle, mitosis, somatic cell division.